All right, here we go. So on the online lecture, I haven't looked at it in a couple of semesters, so I don't remember exactly what all's there, but I think there's several more of the whole three-point map cross example kind of thing. So we're going to kind of move on from that. You got it, right? Yeah, you can use 3 genes, map them, great. We hardly do that ever anymore. You also can't do that with people because you can't really easily set up matings, and you're only looking at the offspring that are available, which are generally fewer in number, 2 or 3 per family. So it's a little more tricky to map genes in organisms like us because of that. So we've tried to come up with other tricks to do it. And one trick is, could you just take all the human chromosomes and put them in a different cell? And what happens is you create something called a somatic cell hybrid. So what's done is basically human cells that are in culture, and these are generally cells that were taken from like a biopsy or something. They're maybe from a tumor or something like that, and then they're cultured in cell culture. And this has been done for a lot of different critters. So one of the early experiments, they took human cells and rodent cells from a hamster and caused them to fuse together. And you can chemically do that with cells. And there's a lot of overlap in the genes because we're not different that much from hamsters. And so a lot of the chromosomes get lost. So you sometimes lose human chromosomes, you sometimes lose hamster chromosomes, and eventually it sort of whittles itself down to have one set of everything that it needs to basically stay alive in cell culture. It's not going to build a hamster, it's not going to build a human, but it's going to live in cell culture. And so those were done, and you created a whole bunch of different lines of these cells, and each line of cells, a cell line, would have a different set of chromosomes. So they wouldn't all be the same. They would have lost different human chromosomes, lost different hamster chromosomes. They'd have some combo, and you could determine which chromosomes were human and which chromosomes were hamsters because of the staining patterns. And so you could then use those and try to detect the DNA sequences of your gene and see if your gene was there. And you kind of use a process of elimination to see if, chromosome 1 is present and my gene is present. If every time chromosome 1 is present and my gene is present, then maybe your gene is on chromosome 1. So it's not a real fine scale mapping. It's sort of saying, yeah, we can tell that this gene is on that chromosome, maybe on one end of the chromosome, but you're not going to know exactly on that chromosome where the gene is. But it gets you at least to a chromosome. And so that's done by doing an experiment that's called a hybrid panel. And so essentially there's several different ways of doing it nowadays. Most probably they would do it by PCR, which is the technique that we would use for a lot of DNA analysis like that. In the old days, they did it by hybridizing molecules of DNA, and that's kind of how it got the name hybrid panel. And it's also from cells that have been fused together, which are hybrids. So that word hybrid comes into play a lot. And so here's just a little baby example. If you had 3 different cell lines numbered here, and you had in these cell lines, you had 1 through 7 of the human chromosomes represented, okay? And you're looking for 4 different genes, then you just sort of do a process of elimination, okay? So if we want to say, where is gene A, okay? Well, it's not present on any of those 4 chromosomes because detecting gene A gave us a negative signal, and those were the 4 chromosomes that were present in this cell. So we know it's not on chromosomes 1, 2, 3, or 4. So that's a process of elimination. You just eliminated 4 chromosomes. You know it's not on one of those 4, right? So the game is kind of figuring out where it isn't as well as where it is. Does that make sense? So now you look at this second cell line and you go, oh, it's here. Okay. So it's on 1, 2, 5, or 6. No, because we already said it can't be on 1 or 2. So now we've narrowed it down to being just 5 or 6 because they're the only 2 that are here that are new. And then in the third line, it's present. And you look, well, it can't be 1, can't be 3. It could be 5 or 6. 6 is not here, but 5 is. And you're like, well, it could be 7. Well, no, it couldn't because 7 was absent, but the gene was present, right? So again, you've eliminated 7, so you've narrowed it down now through the process of elimination to being on 5. Where is it on 5? It's on 5. We don't know, right? So it's kind of a rough location to a chromosome. So where is gene C located? You do that. Think through that. Is it on 1, 2, 3, or 4, or 5, or 6, or 7? So it must be on 8. No, because none of these cells have 8. You can't use a negative result. So if you never detect the gene, you can't say it's on a chromosome you didn't look at. You never looked at 8. They put that in there as one of those things that people want to go to. You can't use negative data to support a conclusion. So where is D? On 1, because 1 is the only one present in all 3 cell lines, right? So you use that sort of logical deductive reasoning to sort these things out. Here's a real life example of they look for in several different cell lines. So these are all the numbers of the cell lines. They just have weird random numbers. There's no rhyme or reason to it really. And they looked in hamster DNA and they looked in human DNA for some gene. I don't know what gene it was. It's probably over here somewhere. And they saw that it was present. So each one of these lines, like cell line 803, has these different chromosomes in it. It has 4 and 5 and so on, right? So wherever there's a black thin box or gray box or partial box, it has part of that chromosome, okay? So in their signal here, they found it in 811 and they found it in 937. They're looking for this dark fragment right here of DNA. So what do those cells have? 811 has these 3 chromosomes. 937 has those 6 chromosomes. So which chromosome is it on? It's present in both. So the chromosome must be in both. Which chromosome is it on? 17, right? So again, you just look at it and go, okay, it's in here and it's in here. It's on a chromosome, so it has to be one of the chromosomes that they share. And the only one they share is 17. So you could say this gene is most likely to be on chromosome 17. Could you get a little more fine scale? Could you take a gene and figure out a way to stick it to a chromosome, basically to stick it to itself on the chromosome? Okay, so when you have molecules of DNA, remember that they have the ability to be unwound from each other and then base pair back together. So if you had one molecule of DNA here and you could unwind it, right? And then you had a second molecule of DNA that you could somehow detect, like make it fluorescent, make it so that it shines a fluorescent color, and then let it base pair. So you have that fluorescent. I'll put a thing here indicating that it's fluorescent, right? Sending off light. So that would be called a probe. So you could take the entire chromosomes in the cell, maybe while they're still in the nucleus or while the nucleus is gone, like during mitosis, and open up the DNA. You can do that with heating it, okay, melt it apart, and then allow it to cool down in the presence of a probe. And so the probe will stick to the complementary sequence of the DNA. So it's basically going to take one strand of the DNA and stick it to its other complementary strand. So the probe will find its complementary sequence. Well, if it's fluorescent or radioactive, then you could use that to detect where it's stuck. With fluorescent probes, this is called fluorescence in situ hybridization. It's fluorescent, as you can see, it's glowing, green or red or purple or something. It's hybridization because you're taking one strand of DNA and allowing it to base pair to another strand of DNA, that is your probe, and it's in situ because you did it while the DNA was in the cell, okay? Not outside the cell in a test tube. So we call this FISH, right? FISH, for fluorescent in situ hybridization. That acronym means that's this process. It's called FISH. And so you can see here they have stained several parts of the chromosome simultaneously. The green is sticking to the centromeres and the telomeres. The red is sticking to some gene. The blue is sticking to some other gene. Okay. Over here, they have hybridized two genes. There's one they've stuck with a green probe. You can see it sticks to two chromosomes. Why? What are these two chromosomes? This one and this one. Homologous pair, right, a homologous pair of chromosomes. So of course they've got the same two genes. They've got a gene here and a gene here, the same gene. So the red one's on one end and the green one's on the other end. So this tells you, you can know that's, I don't know what chromosome that is, but let's say it's 11. You go, oh, it's on chromosome 11, right? We could have learned that with the previous technique, right? But we wouldn't know that it's on the tip end of 11. But now this tells us it's on 11 and tells us a little bit better about where on 11 it's located. Here's one where it's only on the very tip end of just this one chromosome pair. Okay. Why are all the chromosomes red? Why are all the chromosomes blue? Well, they've used some other stain to detect the DNA. That one is called DAPI, D-A-P-I, all caps. It's just a dye. I don't remember what it stands for, diamino something something. And that one up there is called propidium iodide, PI. It's just a dye that sticks to DNA. So that's not sticking to any gene, it's just sticking to the chemical of DNA. So it makes everything look red or everything look blue, okay, so you can see it. And then your probe is fluorescing, red or green, red in this case, green in this case, right? They couldn't use red here because they used red to stain the chromosome, right? Over here, they didn't really stain the chromosomes. They just looked for the probe. They just used something to pick up the centromeres. So if you could stain all the chromosomes with probes like that, somebody smart figured out sequences of DNA that were pretty much the same that kind of went all over the chromosome. And there's a lot of that kind of DNA. We sometimes call it repetitive DNA, okay? So that's what they used here. They used a probe, not to gene, but a probe to parts of the chromosomes that are not genes. And they made that probe different colors. There was only so many colors, so they had to reuse them, but they reused them on different sized chromosomes. So, like for example, you've got a purple one here, and you've got another purple one here, but yeah, this is a big purple, that's a little purple. You've got a big blue and a little blue, and you got two different color blues, and you got yellows, right? Big, medium, small, so forth. Okay, so they kind of figured it out, but they can create a map this way, a karyotype. We've looked at them before, but this karyotype is colored. So it's called a spectral karyotype, spectral karyotype, or spectral karyotype, sky. And that allows you to see chromosomes if they've rearranged quite a bit easier. You don't have to be a pro at looking for the little bands. You just look for the colors, right? Sesame Street stuff. So like, look right here. Here's a red one with blue attached. That ain't right. Not supposed to be mixed red and blue. So that means that part of a blue one, that part right there, detached, reattached over here. The red one detached and reattached over here. That's called a translocation. Whoops. That's called a translocation, when chromosomes do that kind of weird thing. So with this kind of analysis, you can do what we call cytogenetic mapping. You can look for problems where the chromosomes have rearranged, or maybe they're missing or something like that, and you can sort of correlate that with a disease or a condition, right? So what you can observe this way, you can observe when there's the wrong number of chromosomes. So aneuploidies, when there's the wrong number, an extra one or missing one. Translocations like this, where chromosomes are mixed and matched inappropriately. Deletions, like if a chunk of a chromosome is missing, you're going to see a short one, right? And so you can see those kinds of things pretty easily with this. And that kind of gives you an idea, using all of the chromosomes, to get a idea about where a gene might be causing a problem, right? So if you had a cancer patient or a bunch of cancer patients and you did these karyotypes on them, and like 9 out of 10 of them had this problem right here, you'd go, maybe that problem is causing the cancer, right? Maybe this rearranged chromosome correlates with the disease. So maybe there's a gene right here that's being messed up, and the messing up of that gene, or maybe 2 or 3 genes, is what's causing the problem with the lymphocytes or something. So you could use that kind of analysis to try to get at where the gene is that's maybe messed up, causing a disease, right? Does that make sense? You're trying to correlate what's wrong with the chromosomes to what's wrong with the phenotype. So here's one. Here's a crazy one. Look at this chromosome. It's got a little bit of this one here. I guess that's number 8 stuck onto 9, and then it's got some of 22 stuck onto 9. Yeah, that person had some issues, and so they said, well, let's look at your chromosomes. And they're like, well, there's your problem. You've got this rearrangement, and there's a bunch of genes on these chromosomes that have been damaged because of the rearrangements. And those genes are now not regulated right, and they're letting cells do the wrong thing, and that's causing tumors and so forth, right? So that's kind of the way you go about it. Now here's kind of a more traditional karyotype, just to give you a side by side, right? So yeah, you can see that this chromosome is jacked up, and you can see this chromosome is jacked up, so you can pretty much make the conclusion that this went here, but you have to look real detailed at those bands to know. So you have to be pretty skilled, and that's a little bit harder to do. Here you go, nope, that's pretty obvious, right? So that just makes it a little handier. With karyotypes, you know, we have talked a little bit about them, maybe, I don't remember if we did or not, but you always write down the number of chromosomes first, like you count 1, 2, 3, 4, 5, right? So there's 46, so that's the right number. That's good. And you write then a comma, and you put the sex chromosomes. So this has 2 X's. So if it had been a male, it would say XY. And then if there's something messed up, you put those things next. So T for translocation, DEL for deletion, right? And then in parentheses, you put the chromosome that's missing or messed up. So in this case, chromosomes 3 and 9 have rearranged. And if you can tell where it is, you can put the part of 3 that is now on 9. So Q22 moved on to the end of 9, which was Q34. But that part is usually not given. Usually you just stop right here with this part right here. Right? That's good enough. So if you see those kinds of things on karyotypes, that's what they're indicating. The total number of chromosomes, the sex chromosomes, and then if something's wrong, that's going to be next. Okay? So if you had an extra chromosome or you're missing a chromosome or you had a translocation or something like that. If you had multiple translocations, they would just be listed one after the other. Like this one would be T8, 9, 22. You just list them in order, numerical order. So 8 and 9 and then 22 all translocated. Here's an example. This is not actually really correct. I know why it's the way it is. Oops, stop. They didn't want to put whether it was a boy that had it or a girl that had it. So there should be sex chromosomes that are missing, right? So let's say it's a boy, 46XY, 5P minus. That means that the 5P, the P arm of chromosome 5 is gone. Okay? They could have written also del, DEL, but they didn't. Here's one, fragile X. This would have been also a male because you wouldn't have this phenotype if it was female, because females have 2 X chromosomes. So you delete part of one X, no problem, usually. So this would have been 46XY, XP minus. So that means the P arm of X is missing. Now, just as an aside, the only chromosomes that can really be lost, that you can lose and still be alive, is the X chromosome. Because guys, how many X chromosomes do we need to be alive? You only need 1, right? How many Y chromosomes do you need to be alive? Ladies, 0, right? Remember that. Do you need a Y chromosome in your life? So the monosomy X is viable. Monosomy Y is normal, quote unquote, for males. And if you lose any other chromosome, that's lethal, embryonic lethal. So the only viable monosomy is XO. And so we write it as XO. And it's got a name, it's Turner syndrome. It has some phenotypic issues and definitely some reproductive and physiological issues. You can have extra chromosomes. You can have an extra 21. You can have as many X's as you want and as many Y's as you want. It doesn't change much. But you only survive with an extra 21, 15, or 18. Anything else extra is lethal. So our chromosome number is pretty tightly regulated. Any differences, and you have quite a bit of messed up physiology. So if parts of chromosomes were missing, could you use that as a strategy for mapping them? And people said, yeah, we can do that. And it's called deletion mapping or deficiency mapping. Deficiency or deletion kind of means the same thing for us. You're basically doing a complementation analysis. You're looking for the failure to complement between a deleted chromosome and a mutant chromosome. Well, they're both mutant. So what do I mean? So one chromosome we might draw like this, where you have the chromosome and then there's a region here that's missing, missing, right? That missing region of that chromosome, so here's the homolog, it's fine, right? So that missing region is called the deletion interval. It's kind of an ironic term, right? It's an oxymoron. You'd say that chromosome has a deletion. How do you have something that you don't have? I mean, right? That kind of thing. But that's what we say, because what else can you say? So what would happen if you mated that organism? So you have this organism with this genotype, but you cross it with an organism that has not a deletion, but a mutation in a gene. Let's say they're homozygous mutant. Here's their 2 chromosomes. And there's a gene right here that is mutated. Okay. Well, if they mated with each other, what would happen? Well, you could make chromosomes that you can make offspring that have this pairing of chromosomes, right? So these have a mutant phenotype. And these have a wild type phenotype because they're offset here, right? But what's the phenotype of this one going to be? Mutant. Okay, so this one is technically a mutant chromosomally, but it has a wild type phenotype. So that's a little different than what we've talked about before, where we would have taken 2 organisms that are totally mutant and mated them. And if you got 2 mutants and you mated them and the babies are mutant, what would you say happened? Complementation or no complementation? No complementation, right? It failed. Here, technically, we're taking 2 mutants. Although one of them has a wild type phenotype, we're taking 2 mutants and mating them and getting mutants, right? So that's failure to complement. Of course, you're also going to get wild types because this chromosome right here could also pair up with the mutant one, right? So these would actually be wild type. But you get some mutants, right? If it had been a true complementation analysis, you would get 0 mutants at all. So you get some mutants, and that says that complementation failed. So that means if you know where this is, you know where that is, and it fails to complement your mutation, now what do you know? Now you know where your mutation is, right? Your mutation must be in that same region, that same interval. So deletion mapping is a way of taking organisms with mutant chromosomes and trying to match up that mutant chromosome with the deletion on another chromosome and saying, I know where the deletion is, I know what parts are missing, so now I know that my gene is in that region. And it gives you another way of kind of trying to figure out where your gene might be. Now, with things like fruit flies or mice or whatever, you know, you could set up matings to do this. So you could create a whole bunch of different mice that have deletions in all their chromosomes. Then you could go make them with any other mouse you wanted to, right? But you can't do that with people. Okay, usually it's not ethical. So you just kind of have to go, well, do people with deletions in their chromosomes already exist? Yes. Do they have diseases? Yes. Could we just match up the deletion with the disease and see if maybe the 2 things correlate? So that's the kind of strategy for people. So for fruit flies, it works like this. This is all these little red lines represent pieces of missing chromosome. So here's the actual chromosome up on top. I know it's not very, it's a little picture that I stretched out, so it sucks. But this represents the whole chromosome, right? So you can kind of get the idea. There's a whole bunch of genes there. Every little black hash mark up there represents a gene. Well, there's a crap ton of genes. And the little red line lines up with that. So if you go from here, I'll go right in this one. If you go from here down to here, well, that was great. If you go from here down to here, that lines up. Here, I'll do a different one. I'll go this way because that'll be better. So that region lines up. So this part right here is gone in that fly, in this line right here. And their name is deficiency 2L ED 793. Great. So you gave them a name. Could be Bob, Hank, whatever. They're missing that piece of their chromosome. Okay. If you have a mutation in this chromosome and you make them with Bob, you're going to have mutant fly babies. And you're going to know that your mutation is in that region. So you could just take any number of flies you want to with mutations and mate them with all of these different fruit flies, because all of these different fruit flies are just kept in a consortium. People take care of them and they just raise them all the time. So you just have to phone up. You don't phone up anymore. You just get on the internet and you go, send me some, deficiency 1437s. And they go, yep, it's in the mail. You get it in a day. You do your mating. A couple of weeks later, you go, oh, my gene was right there because I didn't get any complementation, right? So here's an example. Let me look at the real example here. Here's a real example. So they got this mutation. The mutant was called Spoon. I don't know why. It didn't like forks or something. I don't know. So it's called Spoon. And here's the chromosome. And so they took all of these different fruit flies that had deficiencies all throughout this region of the chromosome. So here's the fruit fly, and here's its normal chromosome, and it's missing this much. It's missing all of those genes right there. You just draw a line straight up. So this fruit fly, DF1JC70, is missing everything from here to here. Okay, it's gone. Your fruit fly has a mutation somewhere, and you make them and you go, did it complement? Did it? Yes. So your gene is not in the same place. So your gene is not over here. Here's one that failed to complement. So guess what? Your gene is in here, but it's not in here because you already ruled that part out. And this one ruled out over here, and this one ruled out all the way over here. So where's your gene? It must be over here somewhere. And this one says it must be over here because you didn't use this part over here. And here's the smallest one. This one right here failed to complement, and it's missing a teeny tiny little bit of the chromosome. So your gene must be in that teeny tiny little bit of the chromosome. So here is that teeny tiny little bit of the chromosome. It has 4 or 5 genes in it. 1, 2, 3, 4. Yeah, there's only 4 lights. 1, 2, 3, 4 genes. Okay. And then you just check to see which one is your gene. Okay, you can identify it. So you've narrowed it down to 4 genes out of 5,000 genes. That's pretty good. Pretty good. You can work on 4 genes. You can't work on 5,000 genes. So they figured out it was actually this one right here. So that's the kind of way deficiency mapping can be used. For people, you can't do that. But you can look at human chromosomes and you can go, yeah, something is definitely missing. Okay, we've got a deletion here. And this person has an extra ear. So maybe this deletion corresponds to ears. Okay, so here's a real life example. These are all patients that have different deletions, different parts of their chromosomes are missing, and they have different related problems called a syndrome. Wilms tumor, aniridia, which is the messed up eyes, genitourinary anomalies like problems with their kidneys or ureters or bladders or genitalia, and sometimes mental retardation. So the syndrome is called WAGR. Wilms tumor is a tumor in the kidney. So you look at these patients and they have, this one has those two things, this one has all four, this one has those three, they have, this one just has aniridia. So you have different parts of this syndrome, and you want to ask, well, how do these chromosomes line up? And so lining up numerous chromosomes like this, you could say, well, this individual, they just have, they're always just given a number or something, right? So you have anonymity. This individual is missing this region right here. And they have Wilms tumor and genitourinary problems. This person is missing only this little part, and they only have aniridia. So maybe the gene that causes the problem with the eye is located here, right? You look for the smallest region that might cause the problem. And so in looking in these kinds of examples, you sort of look for the narrowest window of missing chromosome, and that correlates with the disease. And in fact, the gene for aniridia is, it's called aniridia as a disease, but the gene is actually PAX6. And it's a gene that's found in mice, it's a gene that's found in humans, it's a gene that's found in fruit flies, and it has control over the development of the early eye to make the tissues to build the eye. And if you don't build the early tissues, you can't build the rest of the eye sometimes. And then Wilms tumor, WT, is a gene, it's called WT, Wilms tumor, and it encodes a protein that helps to regulate cell division. And when that protein's mutated, cells can divide unregulatedly and form tumors that can then get mutations and cause cancers. And those two genes were mapped on this. This is actually chromosome 17, and through this study. And in fact, this was done down at MD Anderson Cancer Center because of the Wilms tumor as a cancer, and actually down the hall from where I was in grad school. It's my 15 feet near fame. Here's another example of several different genes located on a chromosome and several different metabolic problems that go on here, and mapping out the genes based on, okay, these patients have these problems and they're missing this region. Okay, so they're missing these two genes, and so on and so forth. So that's kind of how you do it with people, because you can't set up matings with people that you know, okay, hey, guess what? You're missing a piece of chromosome 4. Have we got a deal for you? You know, we need some babies. That doesn't work. So you look for people who come in with the disease. So what that means is a lot of people have deletions in their chromosomes, but it's not part of a gene, and it causes no phenotypic problem. So they're just walking around asymptomatically in their everyday life with a chromosome abnormality. So you really only find the abnormalities that cause a pathology. Okay, so it's really, you're looking at a small fraction of genes and a small fraction of the chromosomes in that way, rather than looking at the entire chromosome. And people wanted to know the entire chromosomes. So that kind of, this kind of stuff was going on in the early 90s. And people said, we really want the whole chromosomes. So we just need to sequence these darn things. We just need to start sequencing, you know, start at one end and sequence the whole chromosomes. And then we'll know where every gene is. We don't have to do all these complicated things. And we can know where genes are that actually don't cause any problems, because nobody ever comes to a doctor's office if they're not having a problem. So that process kicked off mid-90s, and so you started with the Human Genome Project, which finished up in the early 2000s. But and yeah, so now you can go online and you can look up the chromosomes, and you could see, oh, it says there's a gene right here, but it's actually just predicting there's a gene there. You'd actually have to go in and see if that really is a gene. So the only reason that they could do it, though, was because we already knew where a lot of genes already were located. If we didn't have landmarks, you couldn't have made the map. You couldn't have connected all the data of the DNA sequence. So the mapping was an important part to get ultimately what we have now, which is the human genome sequenced on a computer, and you can just go in and search it. It's a beautiful thing. Here's just some more examples. All right, so the last thing that I want to just mention in passing is trying to figure out from like using pedigrees and using the data that can come from them of to map human genes or other genes in organisms who don't have a lot of offspring. And the first thing is you can never be sure of the phase of linkage when you're just looking with 2 genes. You can really only get phase of linkage if you're looking at 3 genes real easily. And so usually with humans, you're never mapping 3 genes. And in fact, you're generally mapping pieces of DNA that aren't even a gene. They're just called a polymorphism. Polymorphism. Almost no pens are moving. So what is a polymorphism? It's a variable form of DNA. It's a variable sequence. And you have some way of detecting it. And you can look at a family. Here's an example. These 2 parents had 2 different sets of polymorphisms. This one had number 2 and 5. This one had 1 and 6. Okay. Bubba had 1 and 2. Well, number 1 obviously came from mom, and number 2 obviously came from dad. Okay. Now, these represent a pair of homologous chromosomes. So these are 2 alleles, right? So Bubba marries Sue Ellen, and she has 3 and 4 as her types, which are different than anything else. So now they have some kids, okay? So 1 and 3s, 2 and 3s, 1 and 4s, 1 and 3s, 2 and 4s, 2 and 3s, 2 and 4s, 2 and 3s. Great. So we can follow those genes, those alleles. Those are just polymorphisms. Now, mom here had some disease. I don't know what it is. Just we colored it in black, so that means she had a disease. Bubba had the disease, and then 4 of his kids had the disease. So how many times she had the disease, he had the disease, he got number 1 from her, is the disease on number 1? Is number 1 allele and the disease, are they linked? Okay, why do you care? I know where number 1 is. I don't know where the disease is. But if I know that number 1 is linked to the disease, I can jump from number 1 to the disease. I can search for it based on that location on the map. It's like, where's Bob? Well, somewhere outside Corsicana, Texas. I don't know exactly where. Where are you going to start? You're going to go to Corsicana and you're going to start searching, right? Because now you have a relative spot for him. So that's what this is. This is a relative last known location, okay? Closest known location. So what we have to determine is, does the disease actually link to P? Okay, so if it's linked to P, then every time you have the disease, you should also have P1. So disease P1, yep. Disease P1, yep. Disease P1, yep. Disease, crap, P2. Is that okay? What happens during meiosis? Can 2 linked genes become unlinked? And if they do, won't they switch alleles? Right? So the disease is actually paired with P2, and P2 was a dad chromosome. So that represents a recombination. What's the most likely original chromosome? Is the disease on P2 or is the disease on P1? How many have the disease and P1 at the same time? Most of them or fewest of them? Most of them. And what happens most of the time with linked genes? Are they inherited together or are they inherited separately? Together. So P1 and the disease went together 3 out of 4 times. That's a 75% like the parents and 25% recombination. So we might say that represents 25 MAP units. We saw 25% recombinance. But this is only 8 kids, and that's a lot of kids for humans, okay? So usually you're only going to have 2 or 3 kids. So how can you really reasonable statistics? Well, here's the same disease in another family. So you follow that family and you go, how many times did the disease go with the parent? Well, unfortunately, you don't know what mama had or grandmama. You don't know. But you know dad had 1 and 2. And how many times do you get the disease? Well, 1, 1, and disease. 1, 1, and disease. 1 and disease. 2 and disease. So you don't know, is this the original and are these the recombinants, or is this the original and that's the recombinant? You always go with the majority being the original, okay? So we have to go both ways though. So if it's linked to 1, then the map distance is 1 out of 8 kids. 1 out of 8 kids showed a recombination. If it is, and it should be 1 to 1, and if it's this, then it's got 2 different possible linkages. And so in one case, it's got a map distance of 1/8 or 12.5 units. In the other case, it's got map distance of 87. So a LOD score is used. So LOD scores are a complicated statistical formula that you plug a bunch of shit into. But one of the pieces of shit is theta. And theta is your 2 guesstimates. So you plug in a low theta and a high theta, and you look to see where it puts you. And then it gives you all the predicteds in between. If the max LOD score, it's called Zmax, is above 3, you go, yep, those genes are linked. Those things are linked. So a LOD score of 3, what does that mean? What does LOD even mean? It means log rhythm. How do you spell rhythm? Logarithm of the odds of linkage. Linkage. So LOD of 3 is logarithm of something. So what do you need to raise 10 to what the log of what gives you is 3. 10 to the 3, right? Log of 1,000 is 3, right? So that means that the odds are 1,000 to 1 that the genes are linked. Okay? We have to have 1,000 to 1 odds before we'll say that the genes are linked. And 1,000 is 10 to the 3. So log of 1,000 is 3. So the LOD score has to be 3 or higher. This one goes almost up to 4, right? So that means there's almost 10,000 to 1 odds that they're linked. That's great, okay? What if the LOD score only goes up to 0? They're not linked. What if the LOD score is negative? Then that means it's much more likely they're unlinked than linked, right? So this sort of thing is LOD score. I want you to know LOD score. What does it mean? It's a measurement of linkage probability for human genetics. Am I going to ask you to calculate one? Of course, because I've not told you the formula nor shown you how to do it. So of course I'm going to ask you. Of course I'm not going to ask you. But what would tell you that genes were linked? What do you need to know? Needs a LOD of 3 or better, right? LOD of 3 or better. So if I said, hey, these 2 genes have a LOD score of 1.7, are they linked? No, we wouldn't say they're linked. All right. So with linkage, with things that we can, do mapping, old school mapping, 3-point crosses, that kind of thing. But nowadays we don't. You're looking at organisms besides fruit flies like we're humans, so we're only usually interested in humans for disease purposes. And so knowing what gene causes what disease was a big problem, trying to figure out what gene it was so you could figure out what the biology behind the disease was. So mapping genes was the first attempt at figuring out where a disease gene was located. So they had a whole lot of strategies: somatic cell hybrids, FISH, LOD scores, deletion mapping, cytogenetic mapping, anything they could try to get a handle on where the gene was reasonably located. At least get a chromosome, better get a region on the chromosome, get the smallest region on the chromosome you could, find something already on the chromosome that links tightly to your gene, start there and look for your gene. Okay, why? Why wouldn't they just find the gene? Because our chromosomes, even though we have 20,000 genes, we have 3 billion base pairs to hold those 20,000 genes. And the average size of a gene is around 20,000 bases. So there's a whole lot of the genome, 90% of it, that is not a gene. So you're looking for not needles in haystacks, you're looking for transparent snippets of clear plastic fishing line in the ocean, okay? It's really hard to find one gene unless you can nail down at least a region on a chromosome where you should focus in on, okay? So mapping was critical to that. Ultimately, mapping allowed for successful sequencing of the genome. No genome was initially sequenced without mapping until in the past 10, 15 years, where people could sort of brute force through computer modeling the genomes of small organisms, microorganisms, and things like that that don't have a lot of junk DNA. But everything else, plants, orangutans, puppies, there's so much junk DNA in most eukaryotic organisms that you can't just sequence their genome and then put it together. You can put together maybe 70% of it, and then the other 30%, who knows where it goes. And that could really be problematic. So, yeah, sure, keep on. So that kind of says what else could we look at for genes? And if we think about bacteria, bacteria have been utilized in genetics. Okay, now bacteria have a couple of things going for them. You can grow a whole lot of them really quickly. Nobody cares if they die accidentally, and they have viruses that infect them, and you can look at those life cycle changes and gene expression changes a lot easier than you can with more complicated organisms. And bacterial genetics really got its start just after the end of World War II, when a lot of physicists who were, I don't know, midlife crisis kind of things, plus, you know, they made an atomic bomb and they killed a lot of people and they were feeling kind of shitty about it. And they said, well, let's get out of the killing people science of physics and get into the killing people science of biology. So they came in, and the biologists were trying to figure out complicated questions, as biologists often want to do. Like, I want to know everything about a dolphin by sitting on a pier. And you're not going to learn everything about a dolphin by sitting on a pier, except that they come up every once in a while and breathe air. And the physicists said, dudes, take it apart. Do what we did with the atom. Go down to the smallest molecules and figure out how things work at the molecular origin, at the molecular level. And they created molecular biology. And now we understand stuff because we actually understand at the molecular level. So bacteria are obviously haploid organisms. They carry around one copy of every gene. We talked about in lab that that's good or bad, depending on kind of which side of the coin. Are you looking for mutations? You can find recessive ones easy. You can identify the recessive loss of function ones, unless it kills it, and then you can't work on it at all. So there's that. Bacteria have a little bit of complicated reproductive abilities. Most of them just do, we call it binary fission. We don't call it mitosis because They don't have cytoskeletal proteins. They don't have a spindle apparatus. They don't have centrioles. They don't have actin. They don't do any of the stuff that cells, eukaryotic cells do. So we'll call it something different, binary fission. But they have to replicate their DNA and then segregate it into the two daughter cells. So they must have a mechanism for segregating the DNA equitably between the daughter cells. And they also have, most of them, an ability to transfer DNA from one cell to another cell. A sexual reproduction, if you will. So an exchange of DNA, an exchange of genes, that's a sexual mode of reproduction, because that's what's really happening during the fusion of two gametes, is the combination of two genomes. Most of the time, plasmids are going to carry around things called, most of the time bacteria are going to carry around things called plasmids, which are just very small. This really isn't to scale. So if it were to scale, a plasmid would be about this big. Right? Because if this is E. coli, the little green chromosome here, this one, that is 4.65 million base pairs. Okay? Plasmids are usually on the order of 10,000 base pairs. So a lot smaller. So plasmids are called extra chromosomal because they're not part of the chromosome. Okay? And bacteria can have many of them at the same time. And those plasmids are generally carrying only a few genes. And those few genes do something very necessary for the bacteria, or else the bacteria would not maintain them. Okay? Because the plasmids are not equally segregated during cell division. So some cells might end up with no plasmid. What happens to that cell if the plasmid is necessary for something? It dies, and then there's no more of that cell. So the only cells left alive are the cells that in fact keep a plasmid. So it's just a random thing. Of course, the more plasmids you have, the more likely it is you keep one or two, right? So usually plasmids exist in multiple copies. Multiple copies per cell. Okay. Now, what kind of transfers can they do? So in the case of sexual reproduction, they do something called conjugation. They come into a direct physical contact with each other. They send the DNA across through a hole that connects them, like a, I don't know, like a, in space shows where they have a link between two spaceships and the astronauts float across in between. That's basically it. The DNA kind of doesn't come in contact with the environment around the cell. It's kind of protected by this tube, but it goes straight across. Another way is through the infection of bacteriophage, which are viruses. Okay, we call them bacteriophage or phage for short. If you're French, you can pronounce it phage. If you're not French and you pronounce it phage, slap yourself. So phage, they can take DNA from one cell and accidentally put it in another cell. They don't do it on purpose. It's not part of their game plan. It just happens accidentally. So it's kind of a random thing, not that useful. And then the last one is called transformation. And this is when the bacteria bring in DNA from outside their environment. We make use of this one. We big time make use of this one. We are always, as molecular biologists, adding genes to organisms, and bacteria is our favorite organism. E. coli is our very, very favorite bacterial organism. And we're always putting DNA into bacteria via plasmids to manipulate that DNA to make lots of copies, because the bacteria divide very quickly. The plasmid is replicated very quickly and in high numbers, and the bacterium can make a lot of DNA in a short period of time And we can physically harvest that DNA from the bacteria and purify it. So we use the process of transformation to put DNA into cells. We'll talk about, if you remember from yesterday in lab, for the Monday folks, we talked about transformation, and you'll talk about it for the rest of you guys today and tomorrow. But that gives us the ability to add DNA to a cell from its outside environment. Again, it's whatever DNA happens to be out there. We could put a specific piece there, great. What if it's not us doing it? What if they just take it up from some other source, like another dead cell, right? That's where you're going to get DNA from, another dead cell. So if that dead cell is the same as you, then you get some of your same DNA. It might have some subtle differences. But what if that dead cell is not you? What if you're a bacterium like E. coli and you kill one of your neighbors, which is a salmonella? Now what can you do? Now you can take up DNA from a totally different species, right? That's something that bacteria can do, no other organisms can really do on purpose, okay? So bacteria can have DNA from anything. Anything in their environment could get taken up into them. DNA is DNA. They don't really care. Can they use it? Maybe, maybe not, right? So if a bacterium takes up human DNA, it won't be able to transcribe it, and it won't be able to probably translate it because there's differences in the regulatory sequences, but it can certainly replicate it. Okay, so we can put human DNA in bacteria and make a crap ton of it and then use that DNA for other purposes, like sequencing it or whatever. So this is a big one, okay, transformation. So conjugation doesn't happen in all species, but a lot. And it's caused because there's a plasmid that specifically causes it to happen, called the F plasmid or F factor. You can use either name. And any strain that has that plasmid, it's called the F, right? This is the F plasmid. So they're called F plus because they have that. So bacterial genes, just a little bit of rundown on gene naming. Bacterial genes almost always have 3-letter name and then a letter that goes with that. Sometimes also numbers, but most of the time letters. And the 3-letter abbreviation has something to do with the function. So like these are all called TRA. That stands, or the TR stands for transfer. And then there's 2 subsets, A and B, and then a bunch of different sub-members with the letters. And then the name tells you nothing usually about what the hell the protein does. So sadly, that doesn't help us much. So like this one, for example, TRA-A, it makes the protein called pilin, and the pilin is the bridge. So when bacteria have a F plasmid, they can make all these proteins that make something called a pilus, and the pilus can connect to a cell as long as that cell doesn't have a pilus. You can make all the jokes you want. So if you have a pilus, you can connect to a cell that doesn't have a pilus, and it's a one-way transfer through the pilus from the host, this is called the donor cell, to the recipient cell, or this is the donor and the host. And so this cell will take one strand of that DNA and it travels, I don't know how it does it, I'm sure somebody does, but it travels through that pilus, which is just basically a big pipeline, over to the other cell. And once it's in the other cell goes, whoa, here's some single-stranded DNA, I better make it double-stranded, because they don't like single-stranded DNA. And they repair it, and by repairing it, they basically just make it a second strand. And now, when it's all said and done, the whole plasmid, a copy of the whole plasmid is now in that cell. And you never lost the plasmid from this cell because you only moved half of this, you only moved one strand. So you just fixed that by making your second strand over here, and voila, now you have 2 cells that can both make pili, and this cell says, dude, I can make my own pilus. Get the hell off me. And they break apart. And that's conjugation. And it only sticks together for about 30 minutes. So this whole thing happens within a very short time frame, just about half an hour. people said, oh, we could use that to our advantage. We could actually take this, let this start happening, and then throw them in a blender, which would bust these apart and see what got transferred. And we could figure out that genes got transferred. The first gene would be the first one on the order here, right? And then if we wait a little longer, the next gene would be coming in, and there'd be 2 genes over here. So we know that this is number 1, this is number 2. So they decided to start doing that. So they mapped the genes on the plasmid, great, but that doesn't tell you anything about the rest of the cells. So then they started using a problem, or what was a problem, but turned out to be something good. They used a factor, or they used a situation when the F plasmid picked up accidentally other genes that weren't originally part of the plasmid. Those are called F prime. Okay, it looks like an apostrophe, but it's pronounced prime, like Optimus, right? Everybody knows. That's how you write Optimus Prime. Okay, so these F prime plasmids would carry an extra gene, and they could find that they could map those, right? Cool. Sometimes they carry 2 genes. Great. We got 1 or 2. E. coli has about 6,000 genes. So that's going to take a while. And then this dude, Luca Cavalli-Sforza, discovered some bacteria that had accidentally gotten the entire F plasmid inserted into their chromosome. Okay. Big chromosomal problem for the F plasmid, because now the F plasmid can't transfer itself to another cell. Instead, the F plasmid tries to transfer the entire frickin' chromosome to another cell. You can't do that in 30 minutes. And so these things will try to conjugate, but they won't successfully produce daughter cells or progeny cells that could keep on conjugating. So it's kind of the end of the line. So these cell lines were called high frequency of recombination, because they would cause the transfer of bacterial genes from one bacterium into another bacterium, and recombination would cause the recipient to change their phenotype. So what they were seeing was they could have a donor cell, an HFR cell, let's say had the resistance to an antibiotic, and they could have a recipient cell that would be killed by that antibiotic. And they could mix them together with no antibiotic. They could mix them together, let them conjugate for a while, and then they could find recipient cells that were now resistant to the antibiotic. So what they knew was antibiotics were getting transferred from the donor to the recipients, but the recipients could not transfer that antibiotic again. So they knew that they lost the ability, because they got the antibiotic came from the host chromosome. So they said, well, we could use that to map the host chromosome genes. And so this was known as interrupted mating mapping, because you're letting the bacteria start mating, and then, you know, Junior runs in the bedroom. Mommy, mommy. So Junior is a blender. So you just throw the bacteria in a blender, turn it on high, and that causes them to pop the conjugation pili apart. And then you look to see what traits have been transferred. Okay, so the cell that is donating is the HFR cell, and the recipient cell is an F- cell, and the cell that's produced is called an X conjugate. Not to be confused with X-con. That's something different. X conjugate. And it'll have a different phenotype. So for example, if you had a donor that had the ability to make proline and eat lactose. And so in bacteria, the gene names, remember, 3 letters. And then if they have that gene, if they have a functional version, they get a plus sign. If they have a mutant version, they get a minus sign. So they don't have two alleles, so we don't need to write slashes or anything. So proline plus, lac plus is the donor, and proline minus, lac minus is the recipient. And so the donors try to mate with the recipients. They transfer some DNA over, right? After a few minutes, if you let this go for just a few minutes, the lac gene comes in, and the lac gene can recombine so that these cells might become lac plus where they used to be lac minus. So this piece of DNA is not repaired because it's not intact. It's just a fragment. And the bacteria make it double-stranded, but it's not a double-stranded circle. It's just a single-stranded, it's just a linear double-stranded molecule, right? And that's, they don't show you that in the book. So what would actually be here in between would be a double-stranded molecule that has the lac plus on it. And then that piece undergoes recombination. And it replaces the DNA that was in that spot, so that you now have lac plus over here, okay? And that's why these cells were called high frequency of recombination. They caused a whole lot of recombination because they never put the whole thing in. If you put the whole thing in, it won't recombine. It would just have two genomes, okay? So just because it puts in a little chunk, it recombines. Not every chunk recombines. Only a certain small percentage of them recombine, okay? But you can detect it. So if you let it go for just a few minutes, you might get lac plus ones. If you let it go a little longer, you transfer in a little bit longer piece, then you might have a double-stranded molecule in here like this. After a long time, your double-stranded molecule would have the proline and the lac on it. And then both pieces, both genes could get stuck in, right? So all you need to do is let it go, put a big beaker together, let it start conjugating, suck some out, throw it in a blender, bzz, put them on petri dishes and go, okay, after 1 minute, the only thing that got transferred was the lactose gene. After 5 minutes, lactose and proline got in. After 6 minutes or 10 minutes, lactose and proline and tryptophan got in. So what are you doing? You're basically saying, who was the first in? Lactose. So lactose is here on the chromosome. Who's the second one in? Proline. So it's after it. What's the distance? What was the distance in before when we mapped genes? Map units, right? That's recombination. We're not looking at recombination as a factor of percentage. We're just looking at yes or no. But what's our unit that changes? Time. So they use time as a distance. So the bacterial genes are mapped in units of minutes. Okay. So this was done by these two people, Eli, who's a man, and Francois. And so they said, look, we could just take a bunch of different strains with a bunch of different genotypes and made them up with a whole bunch of minus strains with a whole bunch of opposite genotypes and just look and see how long it takes for everything to get transferred, okay? And I'll skip to the punchline. There you go. Here's what they found. After a whole bunch of experiments, they found that you would have transfers that would start somewhere and you get 5 or 6 genes transferred, and that'd be as much as you could know. But you pick another strain and it starts in a different place. So if you take the number 1, it started here and it transferred in like these 6 genes. Okay, great. We know who's next to who for there. Well, let's take a different one and it transferred in some different genes. Let's take a third one and it transferred in some different genes. And a fourth one and it transferred in some different genes. And if you just line up, so if the gene came in, let's say you've got a, you're a gene and you're a gene and you're a gene and you're a gene, right? So we'll start over here So you come in, you're at time 2, right? And then you're at time 5, and 7, and 8, right? Now let's say we start here. So you're going to come in at time, where did you come in before? But you didn't start, that's because you started over there. So now you're going to come in at 2. You're going to come in at 4 and 5. But the timing between them is still 1 minute, right? And the timing between them is still 2 minutes. Now let's say we started here. So you come in at 2 minutes, 3 minutes, 5 minutes, 10 minutes, okay? Still the same number of minutes apart, just in a different order. So by doing all that kind of mapping, they found the same gene over and over again, exactly the same distance from its neighbor, but then you just went 1 neighbor further. And then from there, you went 1 neighbor further over from there. And from there, 1 neighbor further over from that, until eventually all the neighbors were known. And the distance in minutes between the neighbors was calculated. And they found out that to get from one side all the way around the ring back to the original one took 101 minutes. And the map says 100, but it's 101 minutes. Okay, and then they said, well, who is the first gene? Totally arbitrary. They just picked threonine A as the first gene, and that became time 0, and then you just count up from there, all the way around the face of the chromosome clock, 101 minutes, till you're back to threonine A again. Okay, so it's pretty simple. All you need to do is just time the entry of the gene. If this gene comes in at 9 minutes, then that's where it is. This one is at 10 minutes, then that's where it is. This one's at 18, this one's at 22, okay? So you just made them, see who goes first, see who goes second, see who goes third, see who goes fourth. That's the order. How long did it take to get there? That's the distance. It's falling off the log easy. All right. Don't fall off any logs.